All right, let's go over Chapter 12, Nervous System. This is kind of going to be our first look at what's going to be the rest of the semester. We're going to be talking about the nervous system in a lot more detail over the upcoming weeks. Okay, so general functions of the nervous system. The nervous system is going to be your communication and control center, okay? So it's going to collect information. It's got receptors that are going to detect stimuli and send sensory signals to the spinal cord and brain. Then the brain and the spinal cord are going to process and evaluate that information. They're going to determine what their response should be to the sensory input, okay? Then they're going to, the brain and spinal cord are going to initiate the response to information. So they're going to send output via nerves to effectors, either muscles or glands. How is the nervous system organized structurally? Well, you've got the central nervous system and the peripheral nervous system. The central nervous system is going to be your brain and your spinal cord. The peripheral nervous system is going to be the nerves that run along, as well as their ganglia, which is the clusters of the cell bodies along the nerves, okay? However, there are two ways to functionally divide the nervous system. You can have the sensory nervous system or the afferent nervous system, and you can have the motor nervous system or the efferent nervous system, okay? The sensory nervous system is going to receive sensory information from receptors and then transmit it to the central nervous system. Somatic sensory system detects stimuli that you can consciously perceive, whereas the visceral sensory system is going to detect stimuli that you typically do not perceive. So, for example, signals from the heart or from the kidneys, okay? The motor nervous system, this is going to initiate motor output and transmit it from the CNS to the effectors. So you have the somatic motor system and the autonomic motor system, or the visceral motor system. The somatic motor system is going to send voluntary signals to skeletal muscles. The autonomic motor system is going to send involuntary commands to the heart, smooth muscle, and glands. And this, the autonomic motor system has sympathetic and parasympathetic divisions. Okay, so the autonomic motor system is going to be what sends a signal to your heart to increase heart rate when you see a bear in the woods, okay? And then your sympathetic and parasympathetic branches. Sympathetic is going to be your fight or flight response. So, for example, when you see that bear in the woods, your heart rate's going to increase. The bronchioles of your lungs are going to dilate so that you can take in big, deep breaths for when you have to run away from the bear. Those are all going to be examples of sympathetic output. Parasympathetic, on the other hand, is the rest and digest branch. This is what kicks in after you've had a big meal and you need to just rest and pack away those calories, okay? And we'll talk about all of that in detail when we get to it. All right, so this is just showing the functional organization of the nervous system. So let's say that you have an impulse come in from the somatic sensory division. This would be something that you consciously perceive. So, for example, something cold on the skin. Well, that signal is going to travel up through the sensory nervous system, and it's going to go to the central nervous system. At the central nervous system, a decision is going to be made about what to do with that input. And then it's going to send signals to the motor nervous system for output, okay? And let's say the decision is that the area of the skin that's cold, the blood vessels underneath there should constrict. So that's going to go through your autonomic motor branch, okay? This is motor output that is not consciously controlled, or it's involuntarily controlled. The effectors are going to be cardiac muscle, smooth muscle, and glands, okay? So that's just an example of one type of input that comes in and what kind of output goes out. Another example, you might have a visceral sensory input. So this would be sensory input that is not consciously perceived, from receptors of blood vessels and internal organs. Okay, so you might get that input. It goes to the CNS. The CNS decides what to do with the input, and it delivers an output. And remember, you can have an output that's somatic motor that is consciously or voluntarily controlled. And in that case, the effector is going to be skeletal muscle. Okay, so we have some questions here. What function is served by somatic sensory nerves? Well, we said somatic sensory. This is going to be sensory input that is consciously perceived from receptors. Okay, so the function of the somatic sensory nerves is to bring in consciously perceived stimuli, okay, and bring that into the central nervous system. Are nerves located in the central nervous system or the peripheral nervous system? Well, we said that the central nervous system, it just consists of the brain and spinal cord. So that means the peripheral nervous system has the nerves. Is the autonomic nervous system considered a sensory system or a motor system? We go back to this chart here. You can see the autonomic motor system right here. It's considered part of the motor nervous system. And remember, it can be broken down into the sympathetic and the parasympathetic division. Okay, general characteristics of neurons. Neurons are the basic structural unit of the nervous system. Neurons or nerve cells are going to have certain traits. So they're going to be excitable. Excitability, meaning that they have a responsiveness to a stimulus. A stimulus is going to cause a change in the cell's membrane potential, and we'll talk about that here shortly. They exhibit conductivity, which is the ability to propagate an electrical signal. Voltage-gated channels along the membrane are going to open sequentially, and this is essentially what's going to move the signal from point A on the neuron to point B on the neuron. Then they have secretion. They release neurotransmitter in response to conductive activity. This messenger is released from a vesicle to influence a target cell. They also have extreme longevity. The cell can live throughout a person's lifetime, and they are amitotic, which means after fetal development, mitotic activity is lost in the vast majority of neurons. All right, so the parts of a neuron, we'll start here. You have what's called a cell body. It's also referred to as the soma or the perikaryon. It's going to be a plasma membrane enclosing the cytoplasm. It's going to contain the nucleus, and this is going to be the control center of the neuron. Then you have dendrites. These are short, unmyelinated processes branching off of the cell body, and they receive input and transfer it to the cell body. The cell body then decides what to do with that input, whether to send a signal, and if so, what kind of signal, and then it will make the decision, and when it decides to send the signal or the action potential, it will send it down here, which is the axon, the long process emanating from the cell body. This makes contact with other neurons, muscle cells, or glands down here. These are the synaptic knobs, which house synaptic vesicles containing the neurotransmitter. Axons function to conduct action potentials and then are going to release that neurotransmitter at the synaptic knobs. And the neurotransmitter is going to act on a target cell, either another neuron or a muscle or a gland, something like that, and it's going to exert its effects on that target cell. Okay, so a neuron is composed of blank, which transmits signals to the cell body, and a blank, which transmits signals away from the cell body. Well, dendrites are going to be what transmits signals to the cell body. Okay, so there'll be a signal here, the dendrite picks it up, transmits it to the cell body. And then the process that transmits signals away from the cell body is going to be the axon. So dendrites and axon A. Okay, which structure of the nervous system carries action potentials in the direction of a synapse? Well, that's going to be C. A neuron is the structure of the nervous system. We said it was the basic structural unit of the nervous system, and its job is to transmit action potentials towards a synapse. So in addition to propagating the action potential, an axon is also going to move materials to and from the cell body. Okay, so we have anterograde transport and retrograde transport. Anterograde transport is going to be movement from the cell body down the axon. So in this direction, anterograde. And you'll see the substance is moved along a motor protein, which moves along microtubules. Retrograde transport is going to be the opposite direction. It's going to be moving towards the cell body. And this moves used materials from the axon up to the cell body for breakdown and recycling, okay? This can occur by fast axonal transport or slow axonal transport. Okay, so how do we classify neurons? Well, we can classify them structurally or we can classify them functionally. As far as structural classifications go, we classify by number of processes coming off of the soma. So you've got multipolar neurons. These are going to have many dendrites and one axon that extend from the soma. This is going to be your most common type of neuron. You've got bipolar neurons. This is where one dendrite and one axon extend from the soma. These are not found very commonly in the body. The biggest example you're going to find is going to be in the retina of the eye. Unipolar neurons or pseudo-unipolar neurons. This is where one axon extends from the soma. The axon is then going to split into two processes, a peripheral process that then splits into dendrites, and then a central process which leads to synaptic knobs in the central nervous system. And then you can have an anaxonic neuron, which have dendrites but no axons. So here we have an example of the multipolar neuron. You have many, many dendrites coming off of the cell body or the soma and one axon. Here's the bipolar neuron. You have the cell body here and you have two processes extending directly from the cell body, one dendrite and one axon. These are pretty uncommon. You'll find them in the retina of the eye and the olfactory epithelium in the nose. Then you have a unipolar neuron, and you can see here you have a single short axon, single short process coming off, and this is all one axon. You'll have the peripheral process of the axon, which is going to split into dendrites, and then you'll have the central process of the axon, which is going to split into synaptic knobs. And then you have an anaxonic neuron here, which is going to contain dendrites, but no axon. And this is going to be, these are going to primarily be interneurons of the central nervous system. All right, and then we have functional classification, and this is going to be based upon direction of the action potentials relative to the central nervous system. So sensory neurons or afferent neurons, these are going to conduct input from the somatic and visceral receptors to the CNS, okay, so conducting input to the CNS. Most of these are going to be unipolar neurons. Motor neurons, these conduct output from the CNS to the somatic and visceral effectors. Okay, so efferent neurons are going to the effectors. All of these are going to be multipolar neurons. And then you have interneurons or association neurons. These lie entirely within the central nervous system. They're going to receive, process, and integrate information from many other neurons. They form the communication between sensory and motor neurons, and they make up 99% of the body's neurons. Generally, these are multipolar as well. Okay, so a look at the functional classification of neurons. Here you have a sensory neuron. and it's got sensory receptors in the skin. This gets stimulated and it sends an impulse down the sensory neuron towards the central nervous system, okay? Here, it's going to interact with an interneuron, which lies completely within the central nervous system, right? We said the spinal cord is part of the central nervous system. And then the interneuron is going to communicate with the motor neuron and send a signal away from the CNS toward the effector, all right? So this is the efferent neuron, towards an effector, which in this case is skeletal muscle. Okay, the relationship between neurons and nerves. So a nerve, all a nerve is going to be a bundle of axons in the peripheral nervous system. So you take a bunch of neurons, you line their axons up parallel, and then you wrap it in connective tissue wrappings, and there's a nerve. Okay, so the connective tissue wrappings, this is going to sound really familiar to you because it's going to look a lot like what we saw in muscle. You're going to have an epineurium, a perineurium, and an endoneurium. The epineurium is going to enclose the entire nerve, and this is a thick layer of dense, irregular connective tissue. The perineurium is going to wrap fascicles, which is a fascicle is a small bundle of axons in the nerve. This is another layer of dense, irregular connective tissue. And then you're going to have the endoneurium, which is going to wrap an individual axon. And just like we saw in muscle, this is also going to be a delicate layer of areolar connective tissue. This is going to separate and electrically insulate each axon. So this is what it looks like. Here is a fascicle. So surrounding the fascicles, you have the perineurium. Surrounding the perineurium, you have the epineurium. And then in between the individual axons is going to be the endoneurium, all right? And I want you to look at this. Let's just go over what a ganglion is. So here you have a nerve, and a nerve we said is just a bundle of axons parallel to one another. Well, where are the cell bodies to those axons? They are right here, all kind of grouped together, and this grouping of cell bodies is the ganglion. All right, how do we classify nerves? Well, we can classify them structurally or functionally. Structural classification of nerves, you're going to have cranial nerves, which extend from the brain, or you're going to have spinal nerves, which extend from the spinal cord. The functional classification of nerves, you can have sensory nerves, which are going to contain sensory neurons, sending signals to the central nervous system. You can have motor nerves, which contain motor neurons, sending signals away from the central nervous system. Or you can have mixed nerves, which contain both sensory and motor neurons. Most named nerves are going to be mixed nerves. Individual axons in those nerves are only going to transmit one type of information. So here you can see some cranial nerves coming off the brain. Here's your optic nerves. You have several cranial nerves here coming off near the brainstem. And then you have spinal nerves, which come off of the spinal cord. Here and here and here. You have 12 pairs of cranial nerves and 31 pairs of spinal nerves. Okay, so these questions and the rest of the questions throughout the lecture, I want you to spend time going through and making sure you can answer them. If you cannot, email me which question it is that you're struggling with, and maybe I can help illuminate the answer for you, okay? But I want you guys to try to go through and practice on your own. Practice answering these questions, and if you can, you know that you've got a pretty good handle on the material itself. Synapses. So a synapse is going to be a place where a neuron connects to either another neuron or an effector, like a muscle or a gland. The events of synaptic communication. So neurotransmitter molecules are going to be released from vesicles of the synaptic knob into the cleft. The neurotransmitter is going to diffuse across the synaptic cleft and then bind to receptors, postsynaptic receptors, so receptors on either the next neuron or the muscle cell or the gland, okay? Binding of transmitter to those receptors is what's going to initiate the postsynaptic potential. So there's a thing called synaptic delay, and this is going to be the time it takes for all of these events to occur. Okay, so the presynaptic neuron's axon is going to be what produces the signal. So it's going to release neurotransmitters. Those neurotransmitters are going to move across the synaptic cleft, and then they're going to bind to receptors on the dendrites, okay? Most commonly going to receive the signal is the dendrites. Remember that the synaptic cleft is this area here, and it's a small fluid-filled gap between both these neurons. All right, let's talk a little bit about glial cells or neuroglia. So these are the other cells that you'll see in the nervous system. They're non-excitable, unlike neurons. What they do is they provide support to neurons in the CNS and the peripheral nervous system. They are smaller than neurons, but they far outnumber them. They account for about half of the volume of the nervous system. They are capable of mitosis. They protect and nourish neurons, provide physical scaffolding for nervous tissue, help guide migrating neurons during development. They are critical for normal function at neural synapses, and there's 4 types of glial cells we'll talk about: astrocytes, ependymal cells, microglia, and oligodendrocytes. Astrocytes are so named because they are star-shaped cells. They are the most abundant glial cell in the central nervous system. They are going to help form the blood-brain barrier, and what they do is they wrap their feet around brain capillaries, and I'll show you a picture of that in a second. This is going to control which substances can enter the brain's nervous tissue from the blood, okay? They also help maintain the chemical composition of cerebrospinal fluid, and they support neurons, assist in neuronal development, and occupy a space of dying and dead neurons. So here is an astrocyte. You can see it's star-shaped, and it's got these little feet, little projections that it wraps around the capillaries in the brain. And this is going to prevent large molecules, large cells, things like that from crossing over into brain tissue. You also have ependymal cells, which are seen here, and these are going to line the ventricles of the brain and central canal of the spinal cord. Basically what they do is produce and circulate cerebrospinal fluid, or CSF. Next you have oligodendrocytes, which you can see an oligodendrocyte here. These are going to myelinate and insulate central nervous system axons. Okay, and what myelination does is it allows for faster action potential propagation down these axons. Oligodendrocytes are going to be the cells in the central nervous system that myelinate axons. Schwann cells are going to be the cells in the peripheral nervous system that myelinate axons. Finally, you have a microglial cell. These are the phagocytic cells of the CNS. They protect the CNS by engulfing infectious agents and other potential harmful substances. The glial cells of the peripheral nervous system. This is going to include satellite cells and Schwann cells. So satellite cells are going to be arranged around neuronal cell bodies in a ganglion, and they are going to electrically insulate and regulate the exchange of nutrients and wastes. Neurolimocytes, or Schwann cells, I mentioned these already. These wrap around the axons of the peripheral nervous system to form a myelin sheath. And the myelination allows for faster action potential propagation, okay? So faster impulse, the faster an impulse can be sent. So here you have a satellite cell. You can see it is electrically insulating peripheral nervous system cell bodies. And it's going to regulate nutrient and waste exchange for the cell bodies in the ganglia, okay? So here's a neuronal cell body, and then here are the satellite cells. Down here you have a neurolimocyte or Schwann cell, and this is going to myelinate and insulate peripheral nervous system axons, allowing for faster action potential propagation. All right, let's take a minute here to talk about tumors of the central nervous system. So tumor is a neoplasm from unregulated cell growth. Sometimes tumors occur in the central nervous system. A tumor that originates from the brain is going to be a primary brain tumor. But you'll find most of the time tumors in the brain are not primary. They have moved there from another cancer. They've metastasized, okay? But if the tumor does arrive from brain tissue, it's going to be a primary brain a primary brain tumor. Typically, these are going to originate in the supporting tissues. So they're going to come from the glial cells, tissues with the capacity to undergo mitosis. They can also come from the meninges, which are the protective membranes of the CNS, or like I said, the glial cells. Gliomas are glial cell tumors, and these may be relatively benign, but they can be malignant and capable of metastasizing. And one of the gliomas I want to talk to you about is glioblastoma. So this is the most aggressive and most common type of cancer that originates in the brain. It's thought to arise from astrocytes, although we're not sure exactly what cell it arises from. The tumor forms in the cerebral white matter, and it grows quickly. It can become very large before producing symptoms. And you can see right here, you have a large tumor that is crossing the midline of the brain, creating sort of this butterfly look. And then these black areas here are going to be the lateral ventricles of your brain, okay? So the tumor here and here. Okay, myelination. This is the process of wrapping an axon with myelin. And myelin is going to be several layers of the membrane of glial cells. Remember, either oligodendrocytes in the CNS or neurolimocytes in the PNS. They are going to have a high lipid content, which is going to give the axon, the myelin-coated axon, a glossy white appearance, and it insulates the axon. So in the peripheral nervous system, what happens is a neurolimocyte encircles the neuron's axon and wraps it in layers, forming a myelin sheath. So you can see here it's wrapping around and around and around. The neurolimocyte's cytoplasm and nucleus are going to be pushed to the periphery right here, and that's going to form what's called a neurolima. A neurolimocyte can myelinate only 1 millimeter of axon, so several are needed for a single axon. The gaps between the neurolimocytes are going to be nodes of Ranvier or neurofibril nodes. So here you have the neurolimocytes, and you can see the gaps in between where they form myelin. This is going to be the node of Ranvier or the neurofibril node. And this is how it works in the peripheral nervous system. Now, how it works in the central nervous system, one oligodendrocyte can myelinate a millimeter of multiple axons, each at multiple spots. So you can see it's extending processes out to different axons and wrapping at different points along the axon. Again, you're going to have neurofibril nodes or nodes of Ranvier between adjacent wrapped segments. So here is a neurofibril node, here's a neurofibril node, here's a node, etc. There are unmyelinated axons in the peripheral nervous system and the central nervous system. In the peripheral nervous system, they look kind of like this. So you're going to start with a neurolemmocyte that envelops multiple axons. And then see how the axons just kind of spread out, but they're not totally wrapped around by the neurolemmocyte, okay? Not fully ensheathed by it. So they just kind of sit in a depressed portion. In the central nervous system, you're going to have unmyelinated axons that are just simply not associated with oligodendrocytes. All right, let's talk about some demyelinating disorders. We have a permanent one and a temporary one, okay? Permanent one is going to be multiple sclerosis, and this is an autoimmune disorder where the oligodendrocytes are going to be attacked by immune cells. This is going to cause demyelination of neurons in the central nervous system, and it's going to prohibit normal conduction of action potentials along axons. Patients normally start developing symptoms around 18 to 40 years of age. What happens is repeated inflammatory events are going to cause scarring or sclerosis and permanent loss of function in these axons. Symptoms are going to include vision problems like blurry vision, muscle weakness, bladder incontinence, and mood changes, okay? So multiple sclerosis can lead to permanent scarring of the axon and permanent loss of function. Guillain-Barré syndrome, on the other hand, is going to be a loss of myelin from peripheral nerves due to inflammation. Muscle weakness is going to begin in the distal limbs, advancing to the proximal muscles. The exact cause is unknown, but it usually presents symptoms after a flu-like illness. And sometimes you will see Guillain-Barré listed as a possible side effect to vaccinations, okay? But Guillain-Barré, unlike multiple sclerosis, you're going to be able to recover almost all, if not all, function with very little medical intervention.